welcome Professor Karp. We are very pleased to have you as our speaker today. We are super excited to hear your experience about entrepreneurship. It's great to have you here. Before we start, I'd like to spend a few hours talking about Dr. Karp's achievements. I'm just kidding. I'll try to briefly mention some of the achievements that he has made so far through his very successful career. Professor Karp is a Canadian-born biomedical engineer. I'm proud to live in the country that Professor Karp was born in. He is currently a professor of medicine at Harvard Medical School, Brigham and Women's Hospital. He is also the principal faculty at the Harvard Stem Cell Institute, and he is affiliated with the Massachusetts Institute of Technology through Harvard MIT Division of Health Sciences and Technology. He is also an affiliated faculty at the Broad Institute. So that's the first paragraph of the introduction. So I'll move on to the next paragraph. It's like a paper. So he graduated from McGill University in 1999. It's a university that I spent 1.5 years as a postdoctoral fellow. He received a degree in chemical engineering from McGill. Then he joined University of Toronto in and received a PhD in chemical and biomedical engineering under the supervision of the well-known professors Molly Shoykett and John Davis. Later, he received a postdoctoral fellowship from the Natural Sciences and Engineering Research Council of Canada and joined Professor Bob Langer's group using that fellowship. So this is a dream lab that everyone in our community at least would like to join and then spend some time in that. So that was such a great achievement. So far he has published over 125 peer-reviewed papers with more than 20,000 citations. He has given over 300 invited lectures, and oh my God, he has over 100 issued or pending national and international patents. That's pretty amazing. The number of patents that Jeff, you have is more than the number of papers that I have published so far. So that's pretty amazing. And we are not done yet. Dr. Karp is a serial entrepreneur. I mean, he's not only an excellent researcher and scientist, he is also an amazing entrepreneur. He several technologies developed in his lab that have led to multiple products that are currently in development or in the market. And so far, he has launched 8 companies. Oh my God, that's more than the number of papers are published during my PhD. And and I know that even running one company is a lot of work. And then he has raised over $400 million in funding. So technologies that he developed include high-tech skincare, tissue adhesives, 3D printed biomedical devices, immunomodulation materials, small molecule regenerative therapeutics, and so on. So I can go on and then talk about the technologies that he has developed. But let's just get to the last paragraph of the introduction. Sorry, Jeff. I mean, you have so many achievements, and I have to spend a lot of time on introducing you, right? I don't want to miss anything.
No need to. It's okay.
No, it's a pleasure. So just to wrap up Jeff's long list of achievements, it's worth mentioning that Technology Review listed him in 2008 as one of the top innovators under the age of 35. He also received the Young Innovator Investigator Award in 2011 from the Society of Biomaterials, and also in 2012, the Boston Business Journal profiled him as a champion in healthcare innovation. There are many, many other awards that Jeff received, but I don't want to take too much of his time. And then I welcome you, Jeff, to this e-seminar series. It's a great pleasure to have you with us. And then we are excited to hear your interesting talk today.
Great, thank you so much. How do I show my screen?
So if you go on, there's a panel on top. If you click on your image and then your camera, you will see that there is on top. There's a share screen button where you see the microphone.
Can you see my screen?
Not yet.
Okay. Turn off unregistered. Share. Oh, no, not share.
Do you see the share screen button?
Clip. I have clip moment and I have live. This event is currently live and recording, but I don't have a. Yeah, for some reason it doesn't give me the option of sharing my screen, although maybe if I go to presentation mode. Can you see me at all or the
No,
slides?
Houman, do you have any? So there's a share screen button.
Yeah, there is a share screen on your image, on your video. So it's an arrow to the top, like and then you have to choose which file you want to share, like Zoom. You know, you want to share the whole screen. You want to share the specific file.
I don't see it here. Always on.
So do you see the microphone and the video button?
Mohsen, I think we invited Dr. Karp as the speaker. So, I mean, he's on the screen, so he has this. So.
I think you just. Oh, there we are. Perfect.
Oh, so that's great.
I can't hear any volume from you right now. Let's just see here.
Well, you may not be able to see us. Can you hear us?
Oh, wait, here we are. Share screen. Allow. Okay, there we go.
Yeah, exactly.
Sharing your screen.
We can see your screen, but you cannot see us. That's
I can't hear anyone else's voice. Can you hear me or?
Yes.
Close the message. Just see here. Options. Follow. Help. Options. We got it here.
Jeff, can you hear us?
Okay. The sound is echoed.
Mm-hmm.
Okay. Mic. Oh, here we go. Let's see. Save. I've never used this format. Can you hear me? Can anyone hear me? Yes, or I can't. Oh, you can. Okay. Should I just start? Because I can't hear anybody. I just hear myself. Is that okay? Okay. Okay, please start. All right, here we go. And by the way, if there's an issue, please call me because I won't be able to see any of your texts or anything like that. So I'm just I'll get going. Okay. Perfect. All right. Thank you. And I just want to make sure when I put up my slides, can you see them when I just put that up in presentation mode? Everyone could see that? Yes? Okay, perfect. Okay, here we go. Awesome. All right. Well, good afternoon, everybody, and thank you for tuning in. Wanting to welcome you to my kitchen here, where I spend most of my time these days. And yeah, today what I thought I would do is really just share with you just some of the projects that are going on in my laboratory and the stories behind them. How we've really tried to advance in the lab, where publishing papers is really one of the steps to securing impact. And so really that when I started my lab back in July of 2007, I really wanted to have two major goals. One was to create a training ground where people could come from all over the world and be able to realize a significant amount of their potential. Anyone who joins the lab, I commit to being a mentor for life. And then the second was to maximize the potential for translation. And you know, I have been in a number of environments where there's incredibly successful translation going on. However, I think, you know, doing it yourself is a whole different topic. And I quickly realized coming out of my postdoc that I had no, you know, sort of fundamental training in translation, and that I would need to set up a strategy, a lab strategy, a life strategy to really enable that. So I want to share with you some of the aspects of that and that I've tried to implement along the way. And this is a process that I continue to iterate. It's never complete, always learning new insights and you know, other models that other people are using and trying to borrow bits and pieces to improve our potential to help patients. So with that, I'd like to move to my disclosure statement. And so this is required by my institution. I own equity and consult for several companies. These are companies that have been co-founded based on technologies that I have been developing, mostly in the laboratory. You'll see that there's a lot of variety in the various areas that we've been working on. And, you know, I think this kind of speaks to one of the aspects of the lab that I feel is just really core to my interests and curiosities, which is to really focus on the process of medical problem solving and then trying to apply it to lots of different problems. And I'm always very interested to learn about new problems. In fact, I spend time pretty much every day trying to learn about new problems that we might be able to work on. And often when we start new projects, I'm not qualified at all to be working on a project in that area. And so I've really tried hard to surround myself by experts who I can learn from and who we can create synergies and momentum to move things forward. So the general strategy for the lab, and you know, and again, this is kind of an evolving statement, evolving document, as I try to reflect on the process and continually try to update. But really, at the core of this is to try to bring people to the laboratory who are really driven to change the world, and people who are just really easy to interact with and could be you know, positive contributors to the environment and someone who, you know, me and the other lab members could be inspired by. So that's what really one of the core features that we look out for, is being really careful about who we bring into the lab, and because the environment, the way people interact with each other is you know, the most critical aspect, I think, of any sort of workplace. And then what I try to do is to empower people as much as possible. And so the idea is, you know, how can I and the others in the lab create conditions that are conducive for people to realize their potential. And I think as people start to spread their wings and you know, really gain research confidence, that they're continually empowered with additional circumstances or things that they can be subjected to that will help pull them to the next level. So, for example, I'll just give you one example. I had a very talented PhD student who came to my lab, Ben Mead, several years ago, and he was interested in writing grants. And so in his first year, we worked on writing a number of grant proposals together, and 2 or 3 of them actually were funded. And so, he came to me and said, okay, well, what do we do next? And I said, well, you need to hire a team. So I worked with him to hire 2 or 3 full-time people. And so in his second year of his PhD, he was taking classes, and he had 2 or 3 full-time people in the lab who were reporting directly to him. And I think this really enabled him to spread his wings quickly, and to also quickly, realize a significant aspect of his potential. He was able to learn mentorship skills, and being able to direct a team. And I always feel like it's we should be trying to work on things that are kind of stretching our current skills, really taking us out of our comfort zone. And that's where the greatest learning and insights come from. Another example is when we had to go present to a committee, a board of one of the foundations that had funded us. I knew that if I asked them if Ben could attend, they would say no. So I just brought him into the meeting. I presented the first 3 or 4 slides, and then I handed it over to him. And he presented pretty much the whole presentation. And people later from the board came up to me and were like, who is that? Like, is that a junior faculty, or is he a postdoc? And he was in his second or third year of his PhD. And so I think it's like these types of things that are really important to consider, is how can it's a very active strategy. How can we really try to empower people so that they can continue the steepest possible growth curve and be able to maximize their impact, in their careers. The other thing that I've tried to do is there's a lot of inefficiencies and things, in the in every ecosystem. And one of the things that I realized early on was a critical factor for people to really achieve success and to grow was access to resources. And so I made a point early on to achieve multiple affiliations in Boston. So I have affiliations at the Harvard Stem Cell Institute, the Broad, MIT, the Brigham and Women's Hospital, where my primary affiliation is, and then Harvard Medical School, where my academic appointment goes through, and through MIT was through the Health Sciences and Technology program, HST. So, and that was important because I saw that as we were now able to access core resources, core facilities. It makes collaborations much easier if you're an affiliated faculty there, and so this was just one of the ways to try to provide as much resources to the people in the lab. So whenever we need to access a certain tool or technique, or we need to get advice, the activation energy is much lower. I've also tried to actively populate the lab with people from different disciplines and different cultures. We've had people from over 30 countries in the lab, and typically there's very minimal overlap in the expertise of people in the lab. We've had a gastrointestinal surgeon, a cardiac surgeon, multiple MDs. We've had a dentist. We've had immunologists and various types of biologists and various types of engineers. And the way I kind of see it is, if you have people sitting around a table and we're brainstorming, trying to address a certain hurdle or problem that we're facing, you want everyone to be able to provide a unique perspective and not feel any sort of competition. And when you're sitting around a table and each person can kind of provide a unique perspective and ideas, they immediately feel validated and they're more likely to go all in. And so I found that to be very important. And again, people from different cultures tend to think differently. They have different education systems. They have different problems that affect, the people in their country. And so, all of this can be incredibly helpful to solving problems and becoming aware of new problems. And then, as I mentioned before, it's really, I see the 2 major goals of the lab. One is really professional development of the people who are in the lab and making that a highly active goal, as well as translation is a highly active goal. And I see them as being very synergistic. When you commit to being a true mentor to people, they grow a lot faster, they're more comfortable in the environment, and they're more likely to realize their potential and secure more impact in their lives. So these are really synergistic, but they require active strategies. And you can't just sit back. You actually have to really be thinking about everyone in the lab and interacting with them in ways and providing them with the right opportunities that will maximize their growth. So to maximize translation, as I mentioned before, I have no formal business training, and I realized coming out of my postdoc that I needed to really think critically and experiment with my strategy. And so one of the things that I realized, and I kind of just by observing other approaches and observing kind of and reflecting on how I conducted research in the past, is I think often, we're very quick in the biomedical engineering or bioengineering community to focus on the technologies and really try to push them towards, trying to perfect them and trying to test them. But what I realize is that in some ways, and it's not always the case, but often is to really make it a primary goal at the beginning of any project to assume that the problem we're trying to solve has not been defined very well by others. In fact, assume that the problem has been poorly defined or not defined the right way. And that way, and it may have been defined the right way, but that's just, I think, a good way to start projects. So the focus is more on the problem and less on the technology. And then you put your detective hat on and really, go to town early on in conducting experiments with the primary goal to learn, to gain critical insights. And to me, that's been incredibly helpful, just that simple decision of how to think about projects in the early stages. So rather than sort of saying, oh, we're going to develop a technology and test whether it works or get a proof of concept, it's more about, what can we learn that others have overlooked or not learned yet? And then often what happens is if you focus on that, it uncovers some ideas or ways of thinking that then direct you towards particular technologies that others haven't been directed towards. And it gives you kind of a fresh perspective or a new opportunity to solve that problem. The other is I ask a lot in lab meetings, pretty much every lab meeting, what's the bar that we need to exceed to really electrify the field, to get our colleagues in academia excited, to get investors excited, to get people in the translational community excited? What's really that bar? What's the threshold? And one of the ways to think about that is, what's the best result that anyone has ever achieved in a particular model? And then how much better do we need to do to really secure the impact, to really put this on a translational path, to get others excited? And so, this could be, let's say, if you're working on a cancer model, could be that in a particular model, the best has been 20% survival at 4 months. So like maybe if you go to 30 or 40, that's what's really going to take to then propel a new technology forward and secure the investment and mobilize the people around that concept to then be able to then pull that towards a translational endpoint. Could be what's the what's the longest that the animals have survived, and can we extend that by an additional 2 months or 6 months, or so really thinking critically about what's the best result that anyone has achieved, and then how much further do you need to go. And I found that not only is that extremely important from a translational perspective, but it also helps frame things when you submit to journals, to frame the impact of the project. I think often when we submit papers, we're not necessarily directly targeting the impact that we've generated, but that's what we need to communicate to the editors and the reviewers, to really tell them, like, here's where the field is at, and here's how we've gone beyond it. So this simple question that what's the bar we need to exceed to get others excited is just really has been important to the strategy. The other thing is, I realized coming out of my postdoc that there's that I really, because I didn't have a good sense of business acumen, that one of the ways that I could achieve this was through relationships. And so what I did was I committed to meeting somebody new in the translational ecosystem in Boston literally every 2 or 3 weeks. And I've been doing this maybe for the past, call it, 13, 14 years or so, maybe even longer. And so, actively seeking people out, be it a patent lawyer, a corporate lawyer, CEOs of companies, entrepreneurs, reimbursement, regulatory experts, manufacturing experts, and focusing not just on sort of single meetings, but looking for people that I could form relationships with that would almost form an informal advisory board for my lab. People I could reach out to along the way and ask questions to say, what do you think of these results? Like, do you think that, what application would you focus on if you had this platform technology? Do you think this could be manufactured? What would the clinical trial look like? What would our key comparator be? And in fact, that actually is a very important question to ask early on, because if you know kind of what the clinical trial will look like, even in the initial days or weeks or months of a project, if you can kind of advance forward in your mind and really think about that, you probably want to include that comparator in all of your studies, or in a lot of your studies, moving forward in your research, because this is what's really going to separate interesting data from important data. So the difference for me is interesting is, showing that you get a response. We're 5 times better than the negative control, for example. But important is we're actually, 50% better than the gold standard that's used in the clinic. And that type of control is critical if you really want to focus on translational projects. And so I look at this as kind of applying these filters, early on in projects. Can we, often, a huge percentage of the time, what we develop in the bioengineering community can't be translated because it can't be manufactured. And it's not that it can't be made at large scale per se, but often there's issues with quality control. If there's a lot of steps in making something, a lot of potential things can go wrong. It ends up increasing costs and increasing investment dollars. And so we think about manufacturing early on. We think about clinical trials early on. We try to really think about all the downstream things that we can, and then try to apply them upstream, early in projects to maximize potential for translational success. Also, I mentioned, we kind of have this informal advisory board, so I'm constantly getting feedback. I constantly thinking to myself, we must be missing something. There's a problem with the way we're thinking about this. I think it's easy to kind of drink your own Kool-Aid, and I don't think maybe people aren't familiar with Kool-Aid, but we used to drink a lot of it as kids. A lot of food coloring and a lot of sugar in it, so it looked great and it tasted great. But that saying is really about how we get enamored with what we're working on, and then we lose sight of the potential holes. And so I think I've just learned it's really important to constantly seek advice from external groups and people to help you find the holes and address them early on. And then I think another key element here is really trying to publish in the best journal possible, because that trying to get a press release out on the work, because that can alert people or companies. There's been many times where we have a paper published and then a press release goes out, and then companies contact us, or people in those companies who are interested in hearing more, and that leads to something really important. So I think the more sort of and a lot of this is really focused on, again, kind of maximizing the impact, maximizing, really thinking about that translation, that bar that you need to exceed, and then communicating that so you can secure the impact and get the attention of others. And it's a very active strategy. And then also looking for entrepreneurs. So one of the nice things about sort of meeting people in the community every 2 or 3 weeks over the past 13 or 14 years has been I've gotten to know a lot of CEOs, a lot of entrepreneurs, and actually have relationships with them, where we check in multiple times a year and have conversations, go for coffee, these types of things. So when we're advancing, I can now reach out to people and say, hey, are you interested in being involved? Do you know someone who's interested? Because what I've done in the past and what I've seen others do is kind of wait until you have something ready to be translated, and then you look for a CEO. But the challenge is that you can't, these things are relationship driven, and something that I think we overlook a lot because we're just trying to go make a beeline. But relationships end up being, I think, more important than just finding someone who could do the job and do it well. And it's the relationships that are really going to facilitate and maximize the synergies. And when things, you hit bumps in the road, it's really the relationships that are going to hold things together and maximize potential to overcome those hurdles. So that's kind of general strategy. Lots of other elements that I can talk about. Happy to answer questions, but that's just how I've been thinking about things. So I want to turn to a story from the lab, just to give you an example of this translational strategy. And this is a project that we had started over 10 years ago. Dr. Pedro Del Nido, who's chief of cardiac surgery at Boston Children's Hospital, reached out to me, and he was trying to solve the problem of septal defects. So these are holes in between the chambers of the heart, and a number of kids who are born with congenital heart defects have this type of defect. A certain percentage require surgery. And he said, sometimes I go to suture it and the tissue's so fragile, it just tears. There's devices that work in adults, but you can't just downsize them because these are permanent materials. They don't degrade. And as the patient's heart's growing, it's just going to outgrow the device. So he said, is there any way we can work together to try to solve this? And so we were really interested in working with Dr. Del Nido. And so we started to we'd been developing some adhesives early on and published some papers, and that he actually, saw one of those papers, and that's why he reached out. But as we started advancing, we realized that this was probably the most challenging project we had ever worked on in the lab. To get a tissue adhesive to attach inside a beating heart is no small feat. We have the constant flow of blood, which can cause delamination of the material. Every surface is covered with blood. So how are you going to attach to the tissue and not just the blood? Because if you attach to blood, it's just going to fall off. And then you have like the constant expansion contraction cycles of the heart. So again, could lead to delamination. And so we kept hitting wall after wall. And so what we did is we harnessed a tool that we like to leverage in the lab, which I refer to as bioinspiration. And it's really turning to nature to get new ideas. It's this sense that evolution is the best problem solver, and anything that's alive today, any plant or animal, has overcome insurmountable challenges. And if we sort of take some time to look up the mechanisms that have been elucidated in nature, we may gain ideas that could be incredibly helpful to us. And so what we did is we asked a simple question, which is, well, what creatures exist within dynamic environments where they can actually attach to a surface? And we looked at a bunch of creatures, but we kind of honed in on a couple. One is sandcastle worms that exist in the sea, and they sit on rocks and waves hit them and they remain put. And then the other is slugs and snails. Sometimes you'll see a snail sitting on a leaf and it's raining, and it just stays there, doesn't fall off. Or sometimes you'll see it walking along the ground and there's this kind of goo behind it. So what we realized when we started thinking about these types of creatures collectively is that they have a couple things in common. One is viscous secretions. So anything that's viscous has inherent adhesive interactions with its substrate. So, if I put like honey, for example, on a table and I try to wash it away, it takes a while to wash away. And we thought that would be interesting because we started envisioning we would have a patch to seal this hole, fully biodegradable and elastic, and then we'd have a thin layer of glue on the bottom. And we learned from talking to clinicians that sometimes when they place materials, they don't get it in the right location right away, like a hernia mesh or other types of materials. They need to move it around. So our thought was, well, if we made it viscous, we may be able to enable the clinician to place this adhesive and move it slightly to get it into the right position. And the viscosity of that material would enable them to kind of move it around. The other part was that we noticed that these secretions from these creatures contain hydrophobic agents, and hydrophobic agents can repel water. And so our thought was, well, what if we make this viscous pre-polymer, because we're thinking of making a polymeric-based glue that could be activated. We've done a lot of work with light activatable materials. And so we started thinking, well, if we could make this hydrophobic, then it could repel the blood away from the surface, and that might allow us to get better tissue contact. Now, this was a little bit different from how others were thinking, actually a lot different from how others were thinking about it in the field, because at the time that the sense was, if you're going to develop a tissue adhesive, make it hydrophilic to mimic the properties of tissues in the body. But this was kind of telling us, well, maybe we should make it hydrophobic, because a key design criteria for working inside a beating heart is going to be we need to get that blood away. We don't want to adhere to blood. We want to adhere to the tissue. And but that doesn't really solve the issue of how we're going to make this strongly adhesive to tissue. And so for this, we turned to ivy. Walking around Boston, you can see a number of buildings that are covered with ivy. And for anyone who hasn't done this, I suggest you give it a try, which is if you go up and try and pull the ivy off, it's just incredible the amount of force that's required. It's really stuck on there. And the mechanism for this was elucidated about 10 years ago. So ivy has these root hairs, which are like heat-seeking missiles that will go up and down a surface. And when they find a crevice, they'll insert into that crevice, and then they secrete a glue, and then that dries and it kind of shrivels up. So the ivy is able to mechanically interlock with its substrate. And so to achieve very strong level of adhesion. And so we started thinking, well, what if we could get the glue as soon as it attaches to tissue, this viscous hydrophobic prepolymer, what if we could get it to go into the tissue immediately? And then what we'd be able to do is then shine light. Light's able to pass into tissue maybe tens of microns, maybe 50 microns, 100 microns, something in that range. Maybe that would be enough to then shine the light on the material. It would be able to, if we made the patch transparent, we'd be able to then get the light to the glue and then cure it not only on the surface of the tissue, but also in the tissue. And so that was the thought. There are no recipes in the literature for how to do that. So it was really just brute force, lots and lots of experimentation, testing different sort of formulations of the glue. Pardon me. So working with our colleagues at Northeastern, we were able to hand them a sample of this. So what we did is we put the glue onto tissue, onto heart tissue, and then we immediately cured it. We sent it to our colleagues at Northeastern. They did this freeze fracture technique. Here you can see, actually very exciting, here's the adhesive on the top. Here's the collagen fibers in the tissue, and you can see right at the interface that the glue is interdigitated. It's gone in between the collagen fibers to create this mechanical interlock. So it's almost like a tissue-like Velcro interaction that's occurred here. And this was exciting to us because this potentially could be a universal adhesive to attach to potentially any tissue in the body. And importantly, because this material, the precursor, the prepolymer is hydrophobic, this is in many ways kind of agnostic to the environment. It could work inside a beating heart. It could work in the presence of any fluid in the body, and it wouldn't foul the material. And we were able to show that in a number of models. So I want to advance to one of the most challenging experiments that we performed, which is sealing a defect in a rat heart. Anyone who's squeamish at the sight of blood may want to look away because this is an animal experiment. So what we did is we created a hole with a dermal biopsy punch in the rat heart, very large hole. The animal would not survive very long if we left this hole open. We have a purse string suture here that we've used to close that hole just temporarily. We're going to remove it. It's just part of the protocol for this experiment. And then what you're going to see is we're going to come in with a patch, which is transparent. You see high pressure, the heart's beating fast. We have a patch there, a thin layer of glue on the underside. We shine the light through. It goes through the transparent patch. It activates the glue as it's intended. But something goes horribly wrong with this experiment. Kind of worst case scenario. So the patch attached slightly, but it didn't cover the whole hole. We made it too small. And I think we kind of missed placement as well. So we were kind of freaking out, trying to figure out what we can do to salvage this and save the animal. And so we took some glue, we put it onto a spatula and applied it directly to the leak without removing any blood. And if you look carefully, you'll see that it's actually less red here. So the glue is hydrophobic, it repels the blood. Because it's viscous, it stays in place long enough for an extra pulse of light, and we end up with a perfect seal. And we were able to take these animals out 6 months, and they all did fine. So we're pretty excited about that proof of concept experiment. And then we moved into a pig model using a device here that Dr. Del Nido had developed. And what we did is we attached the patch to the end, made a small incision in the myocardium, placed this onto the septum where you typically have these septal defects, and then shine the light for 30 seconds to deploy the patch, and then removed it. And so here you can see 2 pigs. Here's one patch here and another here, 82 beats per minute. So excited to see that the patch was able to attach in the heart, in the presence of blood. And then what we did is we came back about 4 hours later. We added epinephrine to the animal to increase the heart rate to see would the patch detach if under more dynamic conditions. And so here you see we went up to 165 beats per minute, and there's the patch, and there's the patch. So it remained attached inside this beating heart. And then we came back at 24 hours, and you can see the patch here. There's a suture, which is part of the deployment mechanism. So clearly this is not the final experiment. It's really just the beginning, but this was enough of a kind of proof of concept to get us excited, and still a long way to go, and to demonstrate the full degradation, and that we're not going to release segments of this material to cause clots or strokes and things like that. But these sort of initial proof of concept experiments really helped us gain a lot of momentum in moving this forward, and really, I think, helped us overcome some hurdles that were kind of questionable whether we'd be able to overcome them. And the main reason we were able to do that are because of these 2 people here, Nora, who's a cardiac surgeon from Germany, who was co-leading this project. She was in Dr. Del Nido's lab, and Maria, who is a biomaterial scientist who was in my lab. And the 2 of them, Maria's from Portugal, the 2 of them really had a lot of synergy together, like a bioengineer, material scientist working with a clinician. And they were the 2 co-first authors on the paper that we published here. And that synergy was just really, really, I think, important and essential for us to achieve the momentum we did to advance this project, to this stage. So some of you may notice that we had to create a hole to seal a hole, which wouldn't really fly clinically. And so we then teamed up with Connor Walsh at the Wyss Institute and Ellen Roach in his lab, who's now faculty at MIT, to develop an interventional approach. And so I want to play for you this video, which kind of highlights the strategy that we used to try to figure out a way to deploy this patch from a distant blood vessel into the beating heart. So you'll note with this approach that that we actually that there were actually a couple really key things here. One is how do we achieve enough force to really push this tissue adhesive against the tissue? So we needed those kind of double balloon structure and able to do that. So we got really good conformal contact. Another thing was, well, light shines in the direction that you're pointing, but the patch is backwards. It's behind the end of the catheter. And so Ellen had this really great idea to coat the balloon with a metallic coating so that when we shine the light in, it would actually reflect backwards through the patch to activate the glue. And then finally, you'll notice we actually leave a small hole behind, because now how do we get the catheter out? So we spoke to a number of clinicians, including Dr. Del Nido, and they weren't concerned about this. There's actually like a critical size of a hole where it's a problem, but if you leave a small hole, it can really just self-seal in the tissue. And so that's how we were able to advance it. And not only do we have this kind of rendering, video here, but we actually developed this technology and showed that it could work in large animal models. And it's now in the process of moving forward. So based on this technology, we started a company called Tissium, initially called Gecko Biomedical and rebranded as Tissium. And the company achieved a CE mark, which is regulatory approval in Europe for vascular reconstruction. So this is just the glue itself, not the patch, for sealing blood vessels. And this was really seen as an application that was important and something that we could advance quickly, to patients. The application of sealing holes inside a beating heart is something we continue to work on and de-risk. But because this glue we showed could seal the carotid artery of a pig, the aorta of a pig, could attach to gut tissue, we decided to start the company and focus just on the glue as initial kind of translational vertical for this project. And then this technology goes beyond just a glue, because we need to find ways to deliver this in the body. So Tissium has developed a number of approaches. Here on the left, you can see we're spraying it. We've added a blue dye, so you can see where it is. Here in the middle, you can see this is being applied underwater. There was a vacuum-assisted device that was developed to apply that. And then here on the right, you can see this is being placed in a minimally invasive fashion in the body. And so here on the left, again, you can see this light activation. We used UV light in my laboratory. The company uses LED light, which is much safer and easier to deploy. You used to require massive boxes to deliver light in the operating room, but now you can do it just with a pen. So that's been a huge enabler. So I was giving a talk on this technology at the aquarium in Boston, and anyone who's been there will recognize this is actually the penguin exhibit. And so here I am giving this talk on the opening night of this really cool conference, which is called Zoobiquity, which is where human doctors and animal doctors get together and compare notes and learn from each other and gain insights. And I was giving this talk, and everything was going well until the penguins here, which kind of washed out, but you can a little bit see them over here, started getting extremely noisy and very rowdy, actually. And so I couldn't even hear myself speak. So I would stop and turn around and, try to figure out a way to quiet them. And I had to do that many, many times, probably 6, 7, 8 times. So anyhow, that was probably one of the most challenging talks I've ever had to give. I was concerned that no one would hear what I was speaking about. But Dr. Bill Rosenblatt, who at the time was chief of dentistry and craniofacial surgery at an animal hospital in Boston called Angel Memorial, contacted me. He came up to me afterwards and said, I think this could be used in veterinary medicine. And so we kept in touch. And then he contacted me a few months later and he said, I have a dog that has a problem. It's a 10-year-old bulldog, and the dog, I removed a tooth and it left a hole from the oral cavity to the nasal cavity, and it's been getting infected, and I'm really out of options. He had performed 3 separate surgeries to pull tissue over, which is called a flap, where you just cut tissue, pull it over top, and suture it. But because there's so many forces at play, it just tore within 2 or 3 or 4 days after application. So he said, can we use your glue? Can we fill that hole with your glue and pull the tissue flap over top of it after we cure it? And then this would serve as a scaffold for new tissue to form, but would also take on some of the mechanical load in that environment, so it wouldn't all be on the tissue flap. So we were interested to give this a try. Here's what the oral nasal fistula looked like. So you can see it's quite large. It's infected. The animal wasn't eating very well. It was clear that this animal was going to die pretty quickly if we didn't act fast. So I mentioned this to Yuhan Lee in my lab, who's an instructor of medicine, junior faculty. And the next day I come into the lab and I see this, which is a bit shocking because you don't often see like a cow head sitting on the lab bench. But Yuhan had found a way to get a cow head into the lab. And what you can see here is that he applied the glue and he's taken a spatula to it and tried to remove it. And it's remained really strongly adherent. And Yuhan said, "Listen, it's really important to do this because we've never shown this can attach to mucosa in the mouth, and so this was really our proof of concept that this might actually work." And to this day, I have not asked Yuhan where he got the cow head from. I'm actually a little scared to ask him. But anyhow, so we moved on with the experiment. Here we've placed the glue. This has been debrided. I actually have a video on that if anyone wants to see that at some point. It's quite disgusting to remove all the hair and food and stuff that was in here and the infection. We then placed the glue. Here's the tissue flap, which you can see has been used multiple times. We had cut under it, pull it over top, and suture it. Here's what it looks like. And then the animal went away, came back about 3 weeks later, keeping in mind that just the flap had been applied 3 times before and failed within a few days. So Dr. Rosenblad's like wrestling with the animal to try and open, his mouth to get a look, and finally looks inside. And I'll never forget this moment because he was almost crying. It was clear it was a very emotional experience for him. And it hit me on that moment that, this has been his patient for the last 10 years. He had gotten up and planned for surgeries 3 separate times and they had all failed. And this appeared to work. Now, it was only 3 weeks, but this actually ended up working for quite a while. And it worked for a couple years. And so the data we were able to generate from this, I want to share with you, I think is very striking, which is a before and after picture. So here's the animal before the procedure. And you can see he's extremely unhappy. And here's the animal after the procedure. And you can see he's extremely happy. So we're going from unhappy to happy. So this is a remarkable improvement. And this dog's name was Poppy because there's used to be a slugger for the Boston Red Sox called Big Poppy. And I like to, Google everything I hear just out of curiosity. And so I Googled Poppy and I found this image, which, I guess made me figure out why they had called the dog Poppy. And then the local news got wind of this, and we literally had like 3 television channels show up to film Poppy here on the operating table. And so, this was a quite an interesting experience for me. It really opened my eyes to opportunities that I really hadn't imagined. For one, I think there's a lot of opportunity for us to consider that in veterinary medicine, often they reach the extent of what's possible. And even though we're working on experimental approaches in the biomaterials, bioengineering community, some of the things we're working on may actually help animals today. And so I went over to Angel Memorial. I did grand rounds, which means I gave a talk there at like 7 a.m. to a bunch of the doctors, and I keep in touch. And I'd encourage everybody to get in touch with their local, animal hospitals to see if there's opportunities where some of the things you're working on, even if they're not fully tested, may actually be able to help, especially in conditions where the gotten to the extent of what's possible in veterinary medicine. So just a couple other things I want to share with you that we've been working on. One is trying to deliver drugs to the back of the eye. This has been a huge problem if you inject drugs directly into the vitreous, very few of them diffuse to the back of the eye, to the retina, to the photoreceptors, for diseases like diabetic retinopathy or macular degeneration. You can do procedures where you put in probes to the back of the eye and then lift up the retina and inject gene therapy there, although that's extremely limited because it requires a highly skilled surgeon. A lot of things can go wrong, and then you only transfect a very small percentage of the macula. So we were interested in developing a better approach here, and what we did is we tried to look at this from a different angle, which was we looked at the different layers of the eye. And in the eye, you have the sclera, the outer region, and then you have the choroid, which is the next layer. And these are very thin layers. The sclera is about 1 millimeter thick. And in between these layers, you have what's called the suprachoroidal space, which is a potential space. So the two tissues sit against each other. It's almost like a balloon inside of a balloon. And the two balloons are touching. But if you were to put a needle in there, in between, it gets through the first balloon and inject, then you'd be able to inject all the way around. And the tissue would just naturally kind of separate a little bit. And so we developed, recognizing that, this was a space we wanted to access, but we wanted to do it in a way that where we would have great success with clinicians of any skill. We actually developed a needle that automatically stops in between these two layers. And that needle is just based on a force-based interaction. We published a paper on this for anyone who's interested to learn more. Essentially, it's a loss of resistance where when the fluid is going through where the needle's going through the sclera, we have a lot of pressure built up at the tip of the needle. But then when it gets through, all of a sudden the fluid gets released. And we were able to show that we could that we had great success in delivering all kinds of drugs to the back of the eye all the way around. We showed we could do cells and nanoparticles and various contrast agents. And so we formed a company on this called Bullseye Therapeutics, which is now advancing this to the clinic. Pardon me. In addition to this, we've been working on type 2 diabetes, huge societal problem. And one of the really remarkable findings in recent years is that people who are obese, many have type 2 diabetes, and as a procedure for obesity, a number of people get bariatric surgery, in particular gastric bypass. And remarkably, what's happened is that a huge percentage of patients who have type 2, who are obese, after gastric bypass procedures, they don't require any of their type 2 medication anymore, like over 50% of patients. And this is quite remarkable, and there's many papers like this. This is not the only paper. This has been repeated many times. The challenge is that gastric bypass procedure is a very invasive procedure and can only be performed by skilled clinicians, and very few patients qualify. And for those who qualify, few want to get this done because imagine, food will never pass through your stomach again. They kind of reroute your gut. Your stomach is still there to provide various juices, and things can still flow through your GI tract, but food never goes through your GI tract. So we came up with this concept of surgery in a pill, which is, could we develop a pill that you would take before a meal that would transiently coat the same region that you're excluding with gastric bypass procedure, mainly the proximal GI tract? And might this coating be able to physically impair glucose transport, but not be absorbed, so it would just pass through the GI tract? So we developed a material, actually starting from sucralfate. We made some modifications. This is a drug that's been approved for duodenal and gastric ulcers. We were able to modulate that material so that it could adhere to healthy tissue and would actually form a paste, which could then resist nutrient transport through it temporarily. So we published a paper on this a couple of years ago for anyone who's interested in learning more. And we started this company, Ultrix Bio, which is now in the process of bringing this to the clinic. And then finally, just want to share one last story, which is in the area of hearing loss. And so it's a massive societal problem. What's really at the cornerstone of this problem are hair cells in the inner ear. It's not real hair, but these are cells in the cochlea, which can move in response to sound waves, and then the sound converts that signal to an electrical signal and goes to the brain. And when you're exposed to loud noises or just from the aging process or certain drugs, it actually kills these cells and goes away. So you only have 15,000 hair cells per inner ear when you're born, and they never ever regenerate. They just die over time. And we know we had some inspiration from birds and amphibians like frogs and toads because they can restore these hair cells throughout life and restore their hearing. So we knew the biology existed, but in humans, those progenitor cells that exist in the inner ear just remain dormant for life. So what we did is we started in the intestine because there's these cells, LGR5 positive stem cells. We worked out small molecules that can control those stem cells. It actually started off as an intestine project and then morphed into an inner ear hearing loss project. We realized that the progenitor cells in the inner ear are also LGR5 positive. And so on somewhat of a whim, we tested the molecules we had discovered for the intestine to control the LGR5 positive stem cells there. And they ended up working in the inner ear. So we could proliferate those cells, something like 2,000-fold beyond what anyone had shown before, the progenitor cells, and achieve huge numbers of hair cells from those progenitors. And so we started this company called Frequency Therapeutics in 2015. We did a first clinical study in Australia and are currently in a phase 1/2 trial now in the US. Anyone who's interested in learning more about this company can check out the website. The company reported in April of last year that in early part of their trial that a number of patients who had stable hearing loss for months and months, their hearing actually improved in this trial. And so that led to a partnership with Astellas, which is a company based in Japan, also the US. Astellas now has ex-US rights to the hearing loss drugs that are being developed at Frequency. And then this led to an IPO. So we IPO'd the company on the NASDAQ in October. So it's pretty exciting to have the whole team there and really, celebrate. And this also was kind of think of an IPO as a financing event. So its ability to generate more funds to then fuel the clinical programs. So in summary, I talked about a number of technologies we're developing in the lab and some aspects of lab strategy. A couple tools we use quite frequently in the lab. One is bioinspiration. So it's really looking at nature for inspiration, but having a project or having a problem that you encounter first to motivate looking to nature. So we're not trying to just mimic nature. We're actually focused on translation, and then we use bioinspiration as a tool to bring in new ideas. And then radical simplicity, which I actually gave a TEDx talk on. Anyone interested can look that up a couple years ago. This concept is really, I feel it's just so core to the translational motivation and process, especially in academia, which is how can we really think downstream about all the things that we need to think about and then impose them upstream in the most simplest possible way to maximize potential for success. So really thinking about manufacturing and all these things early and making decisions early on that will maximize potential for success later. And most often it's how can we simplify things? Because I think there's this tendency in academia where the more complexity you have in a project and succeed, the higher the impact journal you can get into, but the lower chance of translation. So I think, simplicity is just really a core principle that we turn to all the time. So I just want to acknowledge all of the people in my lab and funding sources, and happy to answer any questions if there's time. So thank you so much.
Thank you so much, Professor Karp. It's always inspiring and encouraging for me, actually, and I think all of the audience to listen to your talk. Every time I listen to your talk, I see new slides on new technology and new companies. So the last time I listened to your talk in Toronto,
I can't hear anything. So
can you hear me?
I'm not sorry about that. Maybe I need to
Okay.
log on as a participant. I could try that if you think.
Yeah.
Or maybe you need to move on. I'm not sure.
So
Share your screen.
okay. So if before you resolve the problem, I'll share some something with the audience, and then we can continue with the question and answer. Okay, so our next speaker is Dr. Amanda Malone. She's talking about diffusion-based extended releasing drug delivery for treatment of osteoarthritis, and then we would like to thank our sponsor TransMedTech and for Biotech to provide supports to have to organize this e-series. So I don't know if
can you see
we
me?
have Dr. Karp.
I can hear you now. Yeah.
Can you hear us?
Okay, good.
Yeah. So I was telling, it's always encouraging and inspiring for everybody to listen to your talk. Like last time I heard your talk, it was in Toronto in Organ on a Chip symposium, and it was. And every time I see that, new slides on new technology and new companies, coming up, every time I see your talk. So before, because I know that your time is limited, so I go to really the Q&A box and start with the great question we got here from Professor Amir Mambachi from Johns Hopkins. He's also an entrepreneur. So he asked you about. He actually thank you for the talk, and then he asked, do you happen to have any advice for assistant professors and aspiring to continue to engage in entrepreneurial ventures? Also, I definitely see impact and merit in it. I'm not sure whether tenure committees would view it the same way. Would be grateful for any hints on balancing the checklists of academia and entrepreneurship.
Sure. So for me, I think I think one aspect of this is just to constantly experiment. So to try to develop an awareness of of others and their strategies, and then try to engage and then try to experiment constantly. Because I think a big part of this is figuring out what aligns with your personal passions, and then what really works for you. For me, one of the things I did was to engage a lot of people in the entrepreneurial ecosystem, and I think that so it's really looking beyond the academic institution and getting out in the community and just developing relationships. And to me, that helps to create gravity. That in everything I do, I'm trying to look for ways to gain momentum towards a translational goal, and so. I found that 100% of the time, the best way to do that is to bring somebody in who has expertise that you don't have or don't have in the team, someone who can provide a fresh perspective and new ideas and different ways of looking at things, can help fill in the gaps or see the holes, help you identify the holes, help you think about downstream barriers or hurdles you might encounter. And so to me, that's been a big part of it. In terms of managing, at the time when I, was doing that, I was having a really hard time during the first 2.5 years of starting my lab. I wrote probably over 100 grant and fellowship proposals, most of them unfunded, because I just was, maybe I was stubborn. I just didn't want to take the advice of others. I wanted to reinvent myself. I didn't want to just focus on things I had done in the past. I wanted to start completely new projects and explore new curiosities. And I just got hammered by reviewers, and my success rate was very low. But then what I realized, it was very frustrating. But then I realized that through that process, I had gained a lot of insights because I got a lot of feedback from the grants. And so now I feel like when I'm writing a grant, I can almost see the 10 people around the table, and I know in my mind, what every single negative thing they're going to say about it, my grant, and about me. So I feel like, part of this is I think to really do this in a major way, you have to have support as well from the chair of your department, and you have to have, be kind of in the right environment. And I think that's also something for people who are looking for faculty positions to consider. Was a big consideration for me is where's a good home for me where I can really fulfill this mission and I'll have the support of others around me. And support might be where, they just leave you alone and let you do what you want to do. Or it maybe they'll kind of, support you in other ways. And so my faculty position's actually through the hospital. So I'm 100%, kind of hard money position. So I have to bring in my entire salary from grants. So it's much riskier kind of faculty type position, but with that, and then I teach, but do less teaching than you typically would do in a traditional academic department. And so that's kind of allowed me to have more time to really spend on these types of things. So I think, if someone's in a department right now, traditional academic department, and they're looking to kind of do more translational things, I think you have to kind of look for things to cut out. And then to make time, because I think, and the biggest thing I would focus on initially would be relationships with people in the ecosystem, as well as looking for people in your community who may be in the same department as you or in academia, who you could try to, become allies with, to try to synergize with, to write grants with. And, I feel like one of the best ways to learn what you want to learn is to submerge yourself in an environment where people are already doing it, or you want to spend the most amount of time possible with the people who are doing it. And so I think those are some of the ways, to go about it. Yeah. And also, people do switch departments and move around. So, thinking about where you're going to get the greatest support is also, I think, important.
Thank you so much. Very inspiring. So the second question coming from Reza Alavi from University of Laval. He's thanking you for the talk, and he asked, could you please elaborate a little bit more on applying simplicity in the research projects?
Sure. So, one example would be, let's say that, and we've done this before. I mean, we've made so many mistakes and gone down, all kinds of dead ends and things like that. And I think one of the things that we used to do a lot of, but not as much anymore, is we used to think that we were doing translation, but we're really doing basic research. And it's important to really be centered on that, because the approach is completely different. If you're going to try to answer a research question and that requires synthesizing a new material that has like 10 steps, like that's totally fine. If you're trying to create a new technology that's going to help patients and it takes 10 steps, that in most cases is not going to be fine, because you're going to create all sorts of manufacturing challenges to do quality control at each of those steps. It's just going to reduce the number of people who will invest. It's going to increase the amount of money you need. So thinking about things like, okay, you've come up with an idea for a technology, but it requires 5 steps. How can you do it in 1 step? How could you do it in 2 steps? How can you reduce the number of steps? Or how can you think about each of those steps and contact manufacturing experts early on about how you could simplify to maximize translation? So it's that kind of, that kind of an approach. I think it's also this approach of really just trying to make a beeline towards translation. So conducting experiments in a way that are focused on gaining insights and getting binary responses, like, yes, this is not working, but then trying to figure it, then having enough data or enough plans in your experiment to figure out why it doesn't work around the mechanism. So I think big part of it is this almost like getting to a regime of simplicity, of not just acting but also thinking about things. And there's always so many things that we can do, but there's very few things we should do. And to me, radical simplicity is about having, gaining experience and insights to figure out what you should do, not the things that you could do. And I talk a lot more about it in this TEDx talk, so you might be of interest to check that out.
I encourage everybody to watch that because it's really inspiring. And the other question, because you said that you hire great people in the lab, so Nagavendra asking, what do you expect when you're looking for a fresh postdoc or graduate student in your lab?
So I didn't mention this before, but one thing is I never hire anyone on my own. I always ask the people in my lab to interview and to come to their own sort of conclusions, and then they can veto me. So like, if I say, oh, I really want to bring this potential candidate to the lab, and they say no, then the answer is no. And the reason is because you want the people in the lab to not feel competitive, with the people who are coming. You want them to be excited. And so to me, that's one of the best filters, at least in my experience, is if the people in the lab, you want them to almost see someone new with a new experience that they could collaborate with and elevate their projects for them to be inspired by. And if you leave it up to the people in the lab to choose who can come to the lab, they'll never pick someone who is going to be competitive. They'll never pick someone who has overlapping expertise. They're always going to pick someone who they think they can get along with, who can elevate their science, who they feel they can, bring more impact. And so that's pretty much, like core to the strategy of what I look for. I just kind of do an initial kind of informal interaction just to get a sense for what really drives people, what they're most excited about. But I turn to the people in the lab to really do, to dig in and help make the final decisions on that.
Wow, very useful, especially for early career researchers and professors. So we got a question also, I have to find it. So you mentioned lots of, successful stories from your lab, but I'm sure that there are lots of failures, as you mentioned. How do you cope with those failures, grant rejections, and how do you cope with that?
Not well, at least initially. I feel like it's kind of like getting punched in the face, right? Because you're kind of putting your best work out there. You're really, you're really excited. You're really trying to think positively, and then you get, So what I try to do is let myself get angry. Try hard not to send any negative emails. Maybe I write them, but I don't send them. And then I think then try not to. I'll try to come back to the summary statement. Not really. I might look through it right away and just kind of get angry, but then just let a little bit of time pass, like a day or two. And usually, if I'm kind of sleeping on it and spending a couple days, you start to come up with ideas of how to improve it. You start to dig in more, engage people in the lab to kind of think about it, maybe contact the program officer and sort of talk it through. And I think through doing that, so giving yourself just a little bit of time to digest it and kind of calm down. You usually can come up with ideas, and you see some of the points that, not always, but you can see some of the points the reviewers made are very valid, and they actually are in general, people are trying to help you improve your grants and do better and come up with better projects and things. Part of it also, I think, is like, it's a game. I think, I think about things like that sometimes, just to manage the psychology of it. And you kind of have to play the game. There's a certain sort of process to getting grants and pleasing reviewers. And so, if you kind of treat it sometimes as a game, you maybe may get less frustrated and more sort of strategic about what you do next.
Great, very inspiring. And this is the last question, I promise, coming from myself. And so you do lots of things. How do you do that? What's the secret sauce?
Well, I'd say number of things. One, I have a very supportive wife, and so that's really kind of front and center to everything. I think, and family, my kids. I also, really try to empower people. Kind of what I've mentioned before is be really careful about, who we bring to the lab, making sure there's a lot of synergy with other people, but then empowering people and not being afraid to kind of let go of the lead on things. So, often postdocs in the lab are leading collaborations, and, I help them prepare in the background, but on calls, I'll let them lead the whole call, any time there's opportunities for them to present. And, so I think it's looking for like win-win opportunities, ways to empower people that will be good for them and allow them to spread their wings more, but then may actually give me a little bit more time to do other things, right? So I think that's, so there's been postdocs in the lab who have led, big industry collaborations, and they're the one leading all the calls. I'm in the background kind of helping them prepare, making sure the slides are good and the order, and and then I'm there for support, but I'm not the one to lead. And so I think, kind of being willing to kind of give up that lead, that front and center position is one of the ways that has been helpful. And then just to bring people to the lab who are able to really grow and achieve like leadership roles has been really critical.
Thank you so much, Jeff. It's always inspiring, as you see in the chat box, and you also answered.
Yeah,
there are lots of comments about your talk. And we really appreciate you that you accept our invitation, and took time to go through all these technologies, and also giving advice to us as early career researchers. So before I close the talk and the session, I would like to encourage everybody. Do you see my? No, you don't see my probably. I have to share.
Okay.
Sorry. Yeah, so I think everybody see our next seminar would be next Wednesday at noon, Montreal time or Eastern time, with Dr. Amanda Malone, and she's the CSO of Apraxia Pharmaceutical, and I encourage everybody also to meet each other again here at the same time, same place, same day. And by that, I would like also to thank our sponsors, TransMedTech Institute, and also for Biotech. And by that, I would close today's seminar, and wish you all the best. And thank you very much, Jeff, again for your great talk.
All right. Thank you so much, everyone, for tuning in. And yeah, great to see you. Okay.
See you. Bye-bye.
Okay.
Thank you, Jeff, for a great talk.
Thank you.
Take care.
Bye, everyone.
Bye.
Bye.
Mohsen, can you close the session?